Hello everybody. My name's Mike Hamblin. For 25 years, I was at the Wellman Center for Medicine in Harvard Medical School, Mass General Hospital. I recently retired and now I'm a visiting professor at the University of Johannesburg. And for the next 20 minutes, I'm going to give you some background introduction to photobiomodulation for brain disorders. So we have a little outline. I'm going to tell you a bit of the history, quite a bit about the mechanisms, because folks are interested in that, and then the clinical studies I've been involved in for PBM of brain disorders. So the history goes way back to 1903, when Niels Finsen won the Nobel Prize for medicine and physiology for using light to treat two diseases, one of which was cutaneous tuberculosis, the other was smallpox. And in his Nobel lecture, he pointed out that he himself suffered from Niemann-Pick disease, and he found that going out in the sunlight gave them a lot of benefits. So this is really when the whole, what came to be PBM started. So over the years since that time, 1903, 1904, light became quite interesting as a therapeutic modality. And since the electric light bulb had just been invented by Edison, as everybody knows, people started to make electric light bulbs and put basically electric light bulbs into cabinets and sort of various bits of apparatus to shine light on the body. And then after that, folks started to really think about sunshine, because obviously sunshine is the best natural source of light. And with folks like Auguste Rollier, who opened clinics that were up in the mountains, in the Alps actually, and you can see lots of folks here outside in the snow, soaking up the sunlight. And Rollier attributed the need to be up in the mountains to the temperature, saying that if you went on the beach and lay in the sun, you only got a hot air bath and not a medical treatment. But in actual fact, I believe it's partly due to the low oxygen tension you get up in the mountains. So fast forward up to the 1960s, when Ted Mayman built the first working laser, the Ruby laser, and 3 years later, Paul McGuff in Boston treated tumors with this Ruby laser. Now, Andre Mester, who was working in Hungary, developed an interest in this laser research, and he got his own Ruby laser. He attempted to repeat Paul McGuff's anti-tumor laser experiment, but this didn't work. And then he said, okay, let's try and see if we can cause skin cancer in mice using this laser. And that failed as well, not surprisingly, because light doesn't cause skin cancer. But he did observe increased hair growth and better wound healing in the mice and the rats, because he had to implant tumors, so there was wounds. So ever since that in the 1960s, this idea of low-level laser therapy, low-intensity laser therapy, low-level this, that, and the other, and various kinds of photostimulation, all sorts of strange names came up. But recently, the field decided to try and unify it by calling it photobiomodulation. A, because nobody quite knew what low meant, and also you're not overstimulating if you've got something like pain, you want to inhibit it. So photobiomodulation is the new consensus term. Now the mechanisms, there's quite a few different mechanisms. The main mechanism revolves around the mitochondria. So the cytochrome C oxidase, which is unit 4 in the mitochondrial respiratory chain, is accepted as a chromophore for red and near infrared light. And the idea is that you get more oxygen consumption, more ATP, the mitochondrial membrane potential goes up. Other things happen, but one important thing is that the mitochondrial metabolism switches from glycolysis back towards oxidative phosphorylation, which is important because it has a few effects on the whole metabolism. You get a lot of signaling happens based on this mitochondrial activity. You get nitric oxide released, you get ATP and cyclic AMP, you get a brief burst of reactive oxygen species. These activate transcription factors, so June Phos is AP1, I kappa B allows NF kappa B to go to the nucleus, and these transcription factors can trigger the expression of over 100 different genes, which is a long-lasting effect. So these proteins that are triggered by these transcription factors will last for hours, days, and even weeks. So a single exposure to light can have long-lasting effects. As I said, this switch of glycolysis to oxidative phosphorylation is important for two reasons. One is that stem cells are activated. Stem cells in their hypoxic niche carry out glycolysis, but when the mitochondria are activated, they need oxygen, so they have to get out of their niche when they can undergo proliferation and differentiation programs. The second effect of this glycolysis to OXPHOS switch is anti-inflammatory. So macrophages have an M1 phenotype and a pro-inflammatory carry out glycolysis. When OXPHOS is activated, they switch to the M2 anti-inflammatory phenotype. And if these happen to be microglia in the brain, they can undergo phagocytosis, for instance, disposing of amyloid plaque that clogs up the brain. There are other signaling pathways that happen when you shine light onto cells, and one of these are the heat-gated TRP ion channels, transient receptor potential ion channels. So blue-green light here can activate opsins, which is probably opsin 4 here. And near-infrared light can activate a conformational change, probably by just basically doing very localized heating of water in the changing the conformation of proteins. And these produce similar results to the mitochondrial activation I spoke of before. You get ROS, you get ATP, but you do get a lot of changes in calcium. There's a third cellular mechanism, and this relies again on this interfacial water, the so-called nanostructured water, and ATP synthase, which is unit 5 in the mitochondrial respiratory chain, is a molecular rotor. So if you decrease the viscosity of the interfacial water, this rotates faster and makes more ATP. So these three mechanisms, the mitochondrial cytochrome C oxidase, the light heat-gated ion channels, and the molecular rotor, all have roughly the same outcomes. Not the same, but similar outcomes. So this was an interesting discovery that didn't happen that long ago. There's a press release in January this year where they discovered that normal blood contains circulating cell-free respiratory competent mitochondria. And that said, does the blood we thought to know so well contain elements that had been undetectable until now? Which is quite interesting how they managed to miss them over hundreds of years. But the idea is that normal blood contains these cell-free mitochondria, and we know that mitochondria are easily activated by light. So the idea is this explains why you can shine light on one part of the body and it can have effects all over the body, a systemic effect, or action at a distance, if you will. Nevertheless, the light does have to get in the body, and only certain wavelengths can penetrate into tissue. And You see here the so-called tissue optical window between 600 and 1200 nanometers, showing that these are the wavelengths that can get in. Another realization that happened too long ago was that there was a biphasic dose response. So if you give a bit of light, you get a positive biological effect. You give a bit more light, it gets better. But if you keep getting more light, it stops getting better. And if you give even more light, the beneficial effect goes away. And if you give a huge amount of light, you could even have a harmful effect. So this has confused a lot of people because they used to do experiments and get negative results. And they couldn't work out whether they weren't giving enough light or they were giving too much light. So it's the idea that the dosimetry is, reasonably important. In humans, it's not actually that critical, but this biphasic dose response does exist. So my laboratory over the years did a lot of experiments in traumatic brain injury. We had funding from the US Department of Defense because TBI was a big concern of the DoD. And, we did lots of experiments in mice and rats with various light sources and various cognitive testing. I won't go through these papers in detail, but we did discover quite a few interesting things about how shining light on the head has beneficial effects. And we've summarized them in this diagram here. So there's a lot of different processes here, and there is positive evidence for all of them, actually. I talked about anti-inflammatory, antioxidant. It's known that light prevents cells from dying, which is important in the brain. Light can cause new blood vessels to grow, which is important if you've had a head injury or a stroke. You need to be able to grow new blood vessels. Reduces swelling, which is important after a head injury. Increases lymphatic drainage, which is important. You get more blood flow, so you get more oxygenation. I believe that these are the important effects down here in the lower right. So you get synaptogenesis, the formation of new connections between existing brain cells. You get neurogenesis, the formation of new brain cells from precursors. And you get these neurotrophins formed, the most important of which is BDNF. So we're not just preserving the brain from dying, but we're actively encouraging the brain to heal itself, which I believe is very important. So getting on to the clinical studies, this is all of photobiomodulation, which can go over the whole body. Again, I won't go into all these in detail, but it really has a very wide spectrum of beneficial effects. Some are relatively superficial, but others are deep inside the body. I just point out this effect on the lungs, which is important in the era of the COVID pandemic, and there are several clinical trials for photobiomodulation to treat COVID-19. Lots of different devices people have used for delivering the light. A lot of these are lasers because it did start off as low-level laser therapy. Whole body light panels are quite popular. There's the NovoThor bed and various big panels. They're flexible devices. You can wrap around various parts of the body, handheld devices. Getting on to the brain, I think Marnie Naser is going to talk in this seminar, so I won't belabor this, but this was one of the early studies showing that 2 case studies using the MEDEX cluster on the head, they had dramatically beneficial effects on 2 people who had chronic head injuries, 7 years or whatever. Another series Marnie did, which was 11 patients with TBI, and out of these 11 patients, 3 did not show a benefit, but the remaining 8 patients showed a dramatic benefit. This was a study that was recently published. It was done at Mass General Hospital, and this was acute head injury, acute moderate traumatic brain injury using a helmet. Here you see the helmet, a real and a sham, and the data here showing that after 2 or 3 treatments, they got treated once a day, but they were only in the hospital usually with mild TBI, mild to moderate, for a few days. But nevertheless, after only a few treatments, they showed a significant benefit. And there was significant benefits also with functional MRI neuroimaging. Again, I think Anita is talking in this seminar. So this was the trial for Alzheimer's disease with Violite in Toronto in Canada. And it's a small trial, but nevertheless, the benefits were dramatic, and both the mini mental state exam and the ADS cog test showed dramatic benefits over the 12 weeks. They fell off after, when the treatment was withdrawn, but since the patients got a take-home device, they approved for a second time, but it's not on the graphs. So there's more trials undergoing because this was quite exciting data, and the results should be available shortly, we think. This was a single case report from Farzad in Iran. Again, somebody with Alzheimer's. He got photobiomodulation on 3 parts of the body, on a sort of helmet on the head, an intranasal device, and a pad on the body. And again, had dramatic improvements on the Montreal assessment, various other assessments. And one interesting thing they found was the sense of smell came back. And you probably know that loss of the sense of smell is a bad sign if you have Alzheimer's. So only a single patient, but quite a dramatic improvement. I think Paolo Casanas, this was Fred Schiffer, who we did the first study on depression and anxiety. So a little LED on the forehead, assessing these folks 2 and 4 weeks later, 10 patients, and they all had a dramatic improvement in depression and anxiety after a single treatment to the forehead with this LED. It faded away after 4 weeks, as you might expect. This was Paolo Casanos' study, where he used a laser on the head and treated them 6 times. Here you go. And following these folks, they had quite a significant improvement in their depression score. This was another study from Fred Schiffer, just recently published, on folks that were addicted to opioids. And he used the same device as in the little LED on the head. The active ones, more than half got better. The sham, less than a fifth got better. So it was a significant p-value. This was just 1 week after treatment. So these are some of the devices that have been used for putting light into the head. Usually near infrared, penetrates better. 810 is quite a popular wavelength. Some use red light. This is the Parkinson's device used in Australia. You can use it on healthy adults. This was older folks, but not diagnosed with any disease. Put the light on the head. And this is one cognitive test, category fluency test, where you have to name like the number of 4-legged animals beginning with B within a certain time. And the ones that got the active treatment could name 3 times as many in the category as the sham group, which is quite a big difference. This was a study again on healthy adults, and what they found here, this is a device that could also measure the cerebral blood flow in the same device that could deliver the near-infrared light to the head, so a dual-purpose device. And they only found minor changes in memory performance in terms of reaction time and what have you. But the very big differences in the cerebral blood flow required to carry out the cognitive tests. So that's why the title of the paper was that the photobiomodulation makes an arduous task less difficult. So you can get the same amount of results with a lot less mental energy, so to speak. This was a study where they were diagnosed with mild cognitive decline. Again, the visual memory and the verbal memory, there were dramatic improvements in some of these. Not every patient improved in every measure, but by and large, the treated ones were a lot bigger than the control ones. So there are a lot of common pathways in all these brain disorders: inflammation, oxidative stress, cytotoxicity, mitochondrial dysfunction, neuronal apoptosis, low BDNF, impaired neurogenesis, hippocampal shrinkage, impaired synaptogenesis. All these have been described as, you know, objective symptoms for various brain disorders, including neurodegenerative, psychiatric, head injuries, strokes, a lot of overlap. But the photobiomodulation can help, I think, in pretty much almost any brain disorder. I mean, it's a bit dangerous to say any, but it's completely harmless. It has no major side effects, no real downside. It's relatively inexpensive. It can be used as a home use device, although a lot of the trials have involved office treatments. You know, folks can be given these helmets or devices to put on their head to use at home. You know, a lot of things like, for instance, if you have a heart attack, you stand a great risk of brain damage. So it makes sense to put some near-infrared light on the head, even when they're trying to get your heart started again, you know, because if they succeed, then you want to minimize your chance of brain damage. A lot of little babies get brain damage through difficult births when they're deprived of oxygen. There's even been some attempts to bring people out of comas by putting light on their heads. Obviously, a lot of psychiatric disorders, a lot of neurodegenerative disorders, and neurodevelopmental disorders, autism and ADHD, all possibly treated with photobiomodulation. So the conclusions: the history is over 100 years old. The mechanisms are becoming understood. We've done a lot of animal studies of TBI, but clinical studies, acute and chronic TBI, Alzheimer's, depression, opioid addiction, cognitive enhancement in healthy adults. And that's it. This was my lab a few years ago when I had a lab, and these are the funding agencies. And thank you for listening.